In this lecture, we will discuss energy and the importance of carbon. All life requires energy, and cells obtain energy in many different forms. The bamboo in this picture captured and converted light energy into chemical energy of sugars. And the panda must consume the bamboo to convert the energy stored in the sugars into different types of chemical energy to fuel his metabolism. But let's start with the basics. The definition of energy is the capacity to do work or supply heat. And there are two main types of energy. Potential energy is the energy of position. The gingerbread man at the top of the stairs has potential energy because gravity is trying to pull him down to a lower position. Kinetic energy is the energy of motion. When the gingerbread man falls down the stairs, he is in motion. The stored potential energy is now put to use to send him plummeting down the stairs. Thermodynamics is the study of energy. If we are studying the energy requirements of a student, we must not only consider the student, or the system, but also their surroundings. Biological systems are open systems because they exchange energy with their environment. The first law of thermodynamics states that energy cannot be created or destroyed. The universe contains a certain amount of energy, and that will never decrease or increase. But energy can be converted into different forms. For example, this is a bioluminescent dinoflagellate, Pyrodinium bahamense, that has converted the chemical energy of its food into light energy. It did not create energy, it merely changed one form of energy into another. The second law states that disorder, or entropy, continuously increases. This is because no energy conversion is 100% efficient. During the conversion process, some of that energy is lost as heat. Notice I said lost, not destroyed. This heat cannot be converted into another type of energy. So unusable heat is building up in the universe with each energy conversion, and this increases entropy. It takes usable energy to maintain order. Now, all organisms obey these laws. As we discussed at the beginning of this presentation, when a plant captures light energy, that energy must be converted. That solar energy is converted into the chemical energy of sugars through photosynthesis. And all living organisms store chemical energy in the form of chemical bonds. To understand how this is accomplished, let's examine how energy plays a part in chemical reactions. There are two main types of chemical reactions, exergonic and endergonic. Exergonic reactions are spontaneous, which means the reactants have enough free energy for the reaction to occur. Free energy is the energy associated with the chemical reaction that can be used to do work. Exergonic reactions release energy, and the products of an exergonic reaction are at a lower energy than the reactants. Remember the bombardier beetle? When the two chemicals in his abdomen come together, an exergonic reaction occurs and energy is released. Much of this energy heats up the liquid product to almost 100 degrees Celsius. Endergonic reactions are not spontaneous. There is not enough free energy to provide the energy required to form a product. So energy from an external source is required for endergonic reactions to occur. The products of endergonic reactions have a higher potential energy than the reactants. The formation of sugars in photosynthesis is an endergonic reaction. Solar energy supplies the extra energy required to form bonds and rearrange carbon dioxide molecules into molecules of sugar. This reaction cannot occur without that extra energy. In fact, these two types of reactions work in concert in cells to power metabolism and maintain life. The energy released from exergonic reactions fuel endergonic reactions. They are coupled, meaning we first must have the energy-releasing exergonic reaction before the energy-requiring endergonic reaction can occur. A large proportion of the reactions are reduction-oxidation reactions, or redox reactions for short. These reactions involve the transfer of electrons. An atom or molecule loses one or more electrons, while another atom or molecule gains these electrons. You can remember this with a mnemonic device. Leo goes grr. Leo stands for lose electrons, oxidation. while GR stands for gain electrons, reduction. Let's look at an example of a redox reaction, the production of glucose. The carbon dioxide molecule is rearranged and the hydrogen atoms are added so that electrons are pulled more closely to the carbon atoms and they are reduced. The water molecule loses the hydrogens and the electrons are pulled away from the atoms, so they are oxidized. But in reactions involving organic molecules, the easiest way to determine where the electrons are going usually involves watching the hydrogens. How many hydrogen atoms does the carbon dioxide have? Yes, none. So the carbon atoms in the 6 carbon dioxides share no bonds with hydrogen. How many hydrogen atoms does the water molecule have? Yes, 2. So the oxygen atoms in the 6 water molecules share bonds with 12 hydrogens. Now to the product side. How many hydrogen atoms does glucose have? Yes, it has 12 hydrogens. So the carbons are now in a molecule that has 12 hydrogens. How many hydrogen atoms does the oxygen molecule have? Yes, the 6 oxygen molecules have no hydrogens. So which molecule gained the hydrogen atoms? Yes, the carbon dioxide molecules were reduced to a glucose molecule. And who lost the hydrogens? Yes, the water molecules were oxidized to oxygen molecules. All life forms are carbon-based. This is due to the bonding characteristics of carbon. You should know by now that metabolism is the sum of all the chemical reactions that occur in an organism. There are 2 main types of metabolism. Catabolism, which is the breakdown of biological molecules. These reactions release energy. And anabolism, which is the building of biological molecules. These reactions require energy. As stated earlier, exergonic and entergonic reactions are coupled in organisms. But we can also say that catabolic reactions and anabolic reactions are coupled in organisms. So how are these reactions coupled? The answer is enzymes. But first, what is an enzyme? Enzymes are biological catalysts that lower the energy required for a chemical reaction to occur. Here we see that the products have a lower energy than the reactants, so that reaction should be spontaneous. However, a certain amount of energy, the activation energy, is needed to get the molecules together and in the correct orientation. Reactants could spend years knocking up against each other before this might happen, but enzymes lower that activation barrier by physically grabbing onto the reactants and forcing them together. Now, when an enzyme is involved in a chemical reaction, we write it above the arrow. In addition, we call the reactants substrates. Let's look at an example of how enzymes do their jobs. Let's look at how the enzyme hexokinase lowers the activation energy of 2 substrates, glucose and ATP, to produce the products glucose 6-phosphate and ADP. Our picture will not show the ATP or ADP. First, which do you think is the enzyme? The enzyme hexokinase is the big blue molecule, and its substrate, glucose, is red, the teeny tiny one. The enzyme has this special area called the active site. This is where the substrate will bind, and the substrate is very attracted to this spot. And when it does bind, the whole enzyme clamps around the substrate, forming the enzyme-substrate complex. The substrate fits in the active site perfectly. The enzyme catalyzes the reaction, and the product is formed. But the product is not the right shape for the active site, and the enzyme spits it out. But the enzyme is not changed in any way. It is ready to catalyze another reaction, and enzymes can catalyze reactions very, very quickly. The rate of product formation for enzymes depends upon how much substrate there is. The more substrate, the more product. That is, until the enzymes have maxed out and are catalyzing the substrate at their fastest rate. Environmental conditions also affect catalytic rates. As temperature increases, enzyme activity increases until it reaches an optimal temperature. Enzyme activity abruptly falls after it exceeds the optimal temperature because the enzyme, being a protein, denatures. This graph shows the activity of 2 enzymes as the temperature changes. Which one of these enzymes do you think is found in a human? Remember that the temperature scale is in Celsius. Right, the gray line. The other is found in a bacteria that live in hot springs. Enzyme activity is also very sensitive to pH. Pepsin is a protein-digesting enzyme produced in the stomach. Trypsin, secreted by the pancreas into the slightly basic small intestine, digests proteins. Which do you think is the pepsin? Yes, it is represented by the orange line, as it is optimal in acidic conditions. Now sometimes enzymes need to be turned off because the product is not needed anymore. Enzymes can be regulated in a couple of different ways. Normally, the substrates bind to the active site, and the reaction is catalyzed, but other molecules can change how enzymes bind. Competitive inhibitors bind directly to the active site of an enzyme. They are shaped similarly to the substrate, but will not produce a product. This reduces the amount of product that can be produced. On the other hand, allosteric inhibitors bind to an area on the enzyme other than the active site. This results in a shape change, and the enzyme's active site is no longer the right shape for the substrate. Again, no product can be made. On the other hand, some enzymes do not work until they are allosterically activated. In this situation, the active site is the wrong shape for the substrate until a molecule binds allosterically. This changes the conformation of the active site, and the substrate can bind and be catalyzed, forming the product.